safe trip back. Well, and thank you also for respecting the template, which is cool from your part. I hope that all the pitch will somehow respect this template so that we compare a little bit. Now, Patrick, if you're okay, we will start with the next session, which is quality by design.
Okay, okay. Now we have the session on quality by design. This is the first time that we organize such a session at within the ETPN annual meeting, because when the organizing committee decided about the program, we considered that this should be presented, especially for all those who are, let's say, developing nanoparticles and or nanoformulations. And I suppose that most of you are involved either in a close way or in a remote way in this kind of activity. So we have selected we have invited 4 speakers. So 2 companies, in very different companies, 1 public organization and 1 research and technology organization. Trying to give you, let's say, different facets about what quality by design means and how it could be implemented and how particles or nanoformulation developers should take this approach into account. So with this introduction, I would like to invite Thierry Bastogne. So Thierry Bastogne is the founder and the CEO of the company CyberNano, and it's a company based in France. And so Thierry will try to educate us, let's say, on quality by design. So 15 plus 5, is that okay for you?
That's okay. Thank you. Thank you, Patrick. So my job, in fact, is to introduce the concept of quality by design. And I think that's the best way to start is to show you an example, just to emphasize the dimension of the problem when you want to design or produce a nanoparticle. So sorry, there is no PDF reader, so I don't know where you can... okay, it's not easy. Oh, that's really a challenge. So this is just an example to say, here's a particle. We have 7 components. Each component could be the shell, for instance. For the shell, you can get different characteristics. For instance, the size, the type of material, so 2 characteristics. And for each characteristic, you can select 3 values to be tested. Small one, medium one, large one. And finally, you've got more than 4 million possibilities of formulation. So now imagine you've got the best one, the best formulation. The second challenge is now to produce it and to get a constant quality of production. Now, if you take the example with 7 or 8 production units, for each unit, you've got different control variables. You can control the temperature, the pressure, the flow rate. For each variable, you can change, you can test different values, 3 in that case. And finally, you've got more than 40 million possibilities to get different conditions of production. So just to illustrate to you that finally, finally you've got a large distribution of possibilities, and so a large amount of risk to take the wrong decision. Decision about 2 questions. The first question is about quality. Is it efficient? 2 questions come after or to give up the process. So it is really 2 important questions. And so now we need a solution to minimize the risk to take the wrong decisions, and we need a solution to speed up, accelerate the process of development. Of course, any prototype can fail, but if we have to fail, we have to fail faster. And quality by design is an approach that has been developed, designed for that, to target that 2 particular objectives. So now just a quick story about quality by design. Quality by design is not new at all. In fact, it has been developed in engineering companies to speed up the design of complex systems in aeronautics, automotive industries. And FDA officials were the first to think that QBD, quality by design, could be adapted to drug development. So it leads to GMP, CGMP, and then finally the Q8 guideline document of the ICH, and then Q9, Q10, Q11 document, and now QED is strongly recommended for any drug development. Not only drug development, also biomedical devices, and also now analytical method development. And if now I have to explain you the main basic components of quality by design, the first step is to define the profile of your nano you want to develop. Generally, you specify the application you want to target, and after you wonder which kind of development of nano I want to target or to reach the application. That's what we call the quality target product profile. The second step now is a really critical step, is about the quality attributes. Quality attribute is defined as a kind of physical, chemical, or biological variable you can measure, and that measurement could be a good picture or image of quality or safety. And that's clearly a characterization problem. Yesterday, Adriel talked about the challenge of characterization. Without good characterization, it is not possible to define a quality attribute. And so you take a risk to finally to be disappointed at the end. So this is a current challenge to get the quality attributes. Then imagine you have specified the quality attribute. The third step is, as I said to the beginning, you've got a lot of parameters. Parameters about the formulation, parameters about the production, too many parameters. So you have to, step by step, eliminate the number of parameters to finally be able to focus on the critical parameters in terms of formulation and in terms of production. It's a risky step because each time you eliminate a variable, you take a risk and you have to assess the risk. So then you've got all the parameters, the quality attributes. You can carry out in vitro, in vivo experiments. You collect the data, and from the data you try to specify this kind of relationship, the cause-effect relationship between the formulation and production parameters and the quality or safety attributes. It's not theoretic, it's kind of data-driven modeling, it's statistics, in a sense that we need this kind of relationship. If you have no relationship like that, you cannot estimate the risk. And the second advantage is you can define what we call the design space. The design space is a kind of region of interest. In the space of formulation parameters, production parameters, you determine a kind of region of interest, and if you are inside this kind of region, you cannot guarantee is too strong, but you can guarantee with an acceptable risk that you control quality and safety. That's the concept of design space. Now, the final step is now, during the production, during the full-scale production, you have to find a kind of control strategy to whatever the disturbance, whatever the change among the the quality of the materials, you have to adapt your control strategy in order to maintain your operating condition into the design space, to be sure that whatever the production, batch to batch, you get a constant quality. That's a challenge, another challenge. Now, behind the main steps of quality by design, clearly, there is a lot of statistics. If you talk about risk, you need a way, you need a method to assess the risk. So you need statistics to estimate the risk, to design the experiment, to control the cost of experimentation, to get the modeling aspect, and for the control activity. So in fact, statistics are everywhere. Just a few comments. I just examined approximately more than 30 papers, recent papers, in which quality by design are clearly indicated. In terms of preclinical nanomedicine, three first conclusions. It's clear that up to now, quality by design is not completely applied. It's not a surprise, because quality by design, if you want to apply strictly quality by design, it could become complex, clearly. And sometimes in papers it is more for formulation, sometimes for production. And the main problem now is we have a large heterogeneity of quality attributes. That's clearly a problem because it's difficult to compare. And so we could, I have no time to develop that kind of conclusion or remarks on the 30 papers. I have to conclude now. And firstly, to sum up, if I have to define like a pitch about quality by design, we can say that QBD is holistic. Holistic means it's a global, horizontal approach engineering from the idea, from the profile of the nano up to the full-scale production. And at different steps of QBD, you have an evaluation of the risk. It could be compared like a statistical go, no go, in fact. That's the first definition. Now, if you wish to apply successfully quality by design, some recommendations. First, it is not a tool. It is not a methodology. It's just a guidance. And so that's not a strict recipe. Clearly, it's a little bit more complex than just a recipe. And in fact, it's a necessary condition, but it's not sufficient. For instance, if there is no cross-collaboration in the partners between in a project, it will not be efficient. I already talked about statistics, and now the main perspectives. Clearly, for nanomedicine, the main blocking point is the characterization problem. We have to be clear and to get a kind of homogeneous vision of which are the main critical quality attributes or safety attributes, clearly. And then we often talk about formulation and production, but we forget one important aspect is the application parameters. If, for instance, I talk about the dose of administration. Dose of administration is not a formulation problem. It is not a production problem. It's the way you use the nanoparticle. So the application parameters has to be taken into account in the process of quality by design. So now it's time, I hope so, the 10 minutes. So thank you for your attention. If you have maybe one question. Oh, maybe it's your Patrick too.
Thank you very much. Thank you very much, Thierry, for this very nice introduction. Question, do you have a question? Yes, Didier.
Yes, thank you for the presentation. In fact, I would just only challenge the last point when you say that dose is not a formulation problem. In fact, it's not a formulation problem when the drug is immediately released, because at the end it's a dose of free drug. But it's definitely a formulation problem for nanotechnology, because what is important is the fraction of dose encapsulated versus the fraction of dose free. And in some project, you can see that according to the dose, the fraction of encapsulated drug may vary. It could be very high in preclinical context because the dose is very high. But finally, the fraction of dose encapsulated in the clinical context may be very low because the drug could be released. So I think specifically for nanotechnology, the dose is a formulation problem.
Yes, maybe it's a bad example, but I think that we have to take into account in the early steps of quality by design the specificity of different applications. According to some application like diagnostic, therapeutics, theranostics, or imaging, for instance, some specific requirements associated with a specific application has to be taken into account because it can interact, for instance, for the formulation. Yeah.
Any other question? Then I have a question. Since you have started a company,
yeah.
I suppose offering this kind of assistance for QBD, can you tell us, of course without disclosing any confidential information, can you tell us, let's say, what kind of requests do you have, let's say, from nanoformulation developers? What kind of help do they ask you for? And because let's say you have nicely presented, let's say, how QBD should be implemented at different stages in formulation and processes. And from your experience, what are, let's say, the picture of the needs for the nanomedicine community?
Maybe 2 or 3 needs. The first is when you try to understand the quality by design, as I said, it could be relatively complex. So there is a need to get something to help to give advice, appropriate way to conduct and to perform the quality by design. The second is generally to save a lot of time with the design of experiments. That's clearly often during many European projects when you have to carry out in vitro and in vivo experiments, that's a cost. And so it's important to get a rational way to organize the in vitro and in vivo experiments. And the last one, I forgot. I forgot the last one, but another project, yes, is to, when you want to collaborate between different European partners with QBD, there is no tool, in fact. So that's why we have developed a kind of internet platform to let the possibility for different European partners to collaborate together in a clean QBD framework. Because if you've got a software, you cannot collaborate. So it's important to innovate on the way to implement the quality by design. So to make it easy, to save time, and to facilitate the application of QBD. This is clearly the need.
Thank you very much, Thierry. And from my personal experience, I would strongly recommend, because we have asked CyberNano to join one of our projects, and we saved a lot of time by this design of experiments. Thierry can testify this. So my recommendation is for those who are developing the nanoparticles, you should contact CyberNano either on a B2B initiative or, let's say, within the framework of a European project. It's worth investing because then you save a lot of time and resources. Thank you very much, Thierry, for this very nice introduction to QBD. And since Thierry mentioned in one of his first slides how QBD has been, let's say, into brackets, invented, and how the regulators have, let's say, adopted this QBD approach. Now we have asked the second speaker, Robert Getzmann from RIVM, okay, thank you, Robert, to make, or even if Robert is not, let's say, a regulator per se, but Robert is in close collaboration with regulators. So Robert will tell us, let's say, a different perspective from the previous speaker, who was a presentation by a company, to say, okay, how we can go maybe even beyond QBD. So we are just, let's say, digesting QBD, but you already go beyond this QBD. So Robert, the floor is yours.
Okay.